I'm going to focus the talk more on applications because I figured Philip would really set me up by giving you the background on how Gaussian processes work. And more or less all the applications that I'm going to show you here today are going to be using a Gaussian process under the hood. So happy to talk or take questions as I go along, especially because a lot of the audience doesn't have a background in neuroscience as far as I know. And yeah, there's technical details that relate to and rhyme with a lot of the things Philip was saying. OK, so let's jump into it. A bunch of the applications that we're interested in over at CCN and in my group and in others is relating how our experience of the world relates to external conditions. And psychologists and neuroscientists study this in many ways, but a really classic paradigm in psychophysics is perceptual paradigms, where you show a stimulus to a human subject and then ask them to tell you something about that stimulus. So for example, I can show you an oriented pattern visual grading and then ask you, is this image tilted to the left or to the right? And if you stare at that and look carefully, you'll probably see that in this case it's tilted slightly to the right. But I can make this task arbitrarily hard, right? I can rotate it so that it's very, very close to vertical, and I can also flash an image like this on the screen for 100 milliseconds or something like this. I can make it arbitrarily hard so that you get it right, no better than chance. And by smoothly varying it around that threshold, I can get you to 60% chance correct, 75% chance correct, et cetera, et cetera. So exploring that threshold of performance is really interesting to psychologists and neuroscientists. And why are we interested in that? We're interested in that because we have this understanding of the brain that when your eye receives photons from a stimulus like this, it's mapped somewhere inside your brain into some sort of what we call an internal representation. And a fundamental fact of neuroscience and psychology is that neural representations are noisy and imperfect. That's why I can make you get this correct only 50% of the time or 60% of the time by varying the difficulty. Okay? So using this internal representation, we then are going to report a behavioral choice. And one of the things that we're interested in is understanding just how noisy this mapping from the external reality to your internal brain state, what is that input-output mapping? And we're going to use in this talk Gaussian processes and a generalization of Gaussian processes called a Wishart process to understand both the mean and stochastic level of noise within that representation. OK, so I'm going to build this up with a really simple cartoon model for what's going on. And this is going to look a little bit like an artificial neural network for those of you who've seen that before. But it actually relates quite closely to a lot of models that neuroscientists care about as well. So remember, I'm set up this task where I'm showing you two sensory stimuli, and I'm asking you to discriminate them somehow. Are they tilted to the left or to the right? And you can have humans do this. You can also set up a binary version of this task, which is so simple that even mice and rats and non-human primates can perform. So then we can actually do experimental manipulations and record neurons in those animals more easily than in humans. So a classic way you would do this is you would set up what's called a go, no-go task, which is press a button when it's tilted to the right and do nothing if it's tilted to the left. OK, so building up a simple cartoon model for how this might work, imagine that I have a brain cell, a neuron, that fires when I'm about to press a button, right? So how would I build a circuit that detects that the stimulus is tilted to the right? And when it does so, I'm going to fire a lot of action potentials and create a motor response. So really simple toy model is that I have sensory neurons that again are receiving these photons, but they're noisy. So they're individual, you can think of uncertain units that are going to get to vote on whether this is being tilted to the left or to the right. And the simplest way, I'm going to call this the number of spikes across these neurons a high-dimensional vector x. And then I'm going to wire them up by, let's just say, linear connections for now to keep it simple, to my output neuron. So this W vector is the weight or a connection between these nodes, just as you would in a very simple multi-layer perceptron or artificial neural net, right? OK, so this is the simplest model that we can start from. And if we simplify this, let's even just take a very simple case where there's only two hidden units, two sensory neurons that are receiving this evidence. Call that x1 and x2, since it's a two-dimensional vector. And now my synaptic weights are w1 and w2. I can show you examples like this. This is simulated data, but if I record real neurons, it doesn't necessarily look that different. So the first neuron, when I show it the go stimulus, it only responds a little bit. And for the no-go stimulus, where I'm supposed to do nothing, it fires more. And neuron 2 is flipped. So this neuron fires more in response to the go stimulus and less in response to the no-go stimulus. So if you show this to people and you don't think about it too hard, and I ask you the question, OK, how should I wire up these two neurons, x1 and x2, to the output neuron in order to get the right response? Your gut reaction is to say, OK, this neuron is negatively correlated with the go stimulus, press the button. And this neuron is positively correlated with the go stimulus. So I want maybe this synaptic weight to be negative, so this is suppressing my urge to press the button, and this one should be positive. So when this neuron fires more, I'm more likely to press the outcome, right? But even just the way that I'm presenting it to you, you can maybe guess what's coming next, is that this intuition, sometimes it's correct, but in this simulated data set, it's absolutely wrong. I'm wondering if anybody actually knows there's a person's name attached to this paradoxical result. This ends up being an example of Simpson's paradox. So what you maybe think is if neuron 1 and neuron 2 are completely independent, one way that I can represent the data is by plotting each trial on, this is x1, number of spikes from neuron 1, x2, number of spikes from neuron 2. Each point is a trial, and I have two distributions. This is my distribution over what the neural responses will be to the go stimulus and the no-go stimulus. And in this case, there's no correlations between the two neurons. And then in this case, your optimal discriminating boundary, which corresponds to your optimal synaptic weights, W1 and W2, is as you would expect. One is positive and one is negative. But Simpson's paradox comes along when I can play with the noise correlations and get your optimal discriminating boundary. Now, both optimal weights are positive excitatory connections. OK? Questions on that? Did everybody follow through? I know you've been sitting in those chairs all day. So, OK. Yes?
Limiting yourself to linear things you can do
Yes.
with the firing rates, right?
Yes.
If I said I actually want to take the product of the two or something, that might be an even better discriminator, right? But you're not allowed to.
Yeah. I was trying to keep, a very simple linear, this would be the optimal linear circuit model to solve this task. And this kind of corresponds exactly to linear discriminant analysis and kind of, machine learning 101 model. But anyway, I'm just walking through this to show you how this maps onto, how a neuroscientist might start thinking about toy circuits that perform these discrimination tasks. And the take-home messages from the beginning of the talk, well, the first is that when I fit an optimal linear model to do a discrimination or a regression, you need to always be careful about interpreting the regression coefficients, because the marginal correlation of each variable with the output variable is not necessarily going to correspond to what the regression coefficient is when you fit it jointly with multiple independent variables. Okay, so that's the first kind of general take-home message that's not even relevant to neuroscience. That's just a general statistical fact. And the second take-home message for the rest of the talk, which is important, is that as neuroscientists, we really care about understanding what is the mean response of a noisy population of neurons. But we also care about measuring what neuroscientists call the noise correlations, or the trial-by-trial variability and correlations among the neurons. Why do we care about that? Well, because you get counterintuitive effects like this depending on what the exact structure of the covariance is. Okay? So for the rest of the talk, I'm going to be just walking through a couple examples where we use Gaussian processes to estimate the mean response, and we're going to use something called a Wishart process to help us model how the covariance changes as a function of the stimulus condition. Okay, so the problem that immediately jumps out at this point is that estimating covariance is very, very challenging in high-dimensional spaces. So imagine, for example, I have a pool of 1,000 neurons that are responding to the sensory stimulus. It's actually, in your real brain, you have billions of these, millions of these neurons. But we can record on the order of 100 to 1,000, and we're interested in estimating correlations. So estimating the mean response is already difficult enough because it's a point in this really high-dimensional firing rate space. That's high-dimensional estimation of a mean vector. Estimating the covariance is even more difficult because I need to estimate what are the axes, principal axes of variation, and what are the widths of each of those axes. So every time I add a new dimension, I'm making my problem much harder and harder. And just to make sure that everybody's following here, in this case, I simplified it down to 2 neurons so that we can visualize it. On this slide, I'm doing something that a neuroscience audience would immediately follow along with, which is, okay, I can only draw this in 3 dimensions, but I want to think of each axis here as the firing rate of an individual neuron. So I'm showing you 3 neurons here, but in reality, this is a much higher dimensional space. And if you count up the number of free parameters you need to estimate, well, to estimate the mean, I need to estimate the mean response for each neuron. So if I have 1,000 neurons, I have 1,000 parameters to estimate the mean. And then I have on the order of 1,000 squared parameters to get all the pairwise correlations, which go into this covariance matrix, which, of course, is one-to-one matched with this high-dimensional ellipsoid. Okay, so this high-dimensional covariance estimation problem is very challenging. It's actually a very hot topic in modern statistics. There's many applications outside of neuroscience as well. But in neuroscience, this is one of the areas that it crops up and we're very interested in. Okay, so in the next part of the talk, I'm going to walk you through our approach to fitting these covariance matrices in high-dimensional space. This is work that was led by a Flatiron research fellow, Amin Najakbash, who just left us for greener pastures, with a lot of help from Isabel Guerin, who's in the audience and still a graduate student in my group. Okay, so just to make this very concrete, I'm going to talk about a data set where we record in motor cortex of a non-human primate that's making point-to-point hand reaches. This is data from a lab that studies brain-computer interfaces with the idea of restoring motor function to people with ALS and other forms of paralysis. So trying to understand the mapping between neural activity in a part of the brain that's related to muscle activity. And what they do is they train monkeys to move their arm in certain directions, and then they measure the neural activity, and they're trying to make a correlation there. Okay? So we're going to look at what's happening in a particular point in time right before the muscle activity starts. It's kind of the initial command. And these are the targets that the monkey's reaching to. So the monkey will always start with his hand in the center of the screen, and it'll make outward reaches to targets on a 2D plane like this. In one condition, the monkey's reaching to a ring. In other cases, it's making reaches to points on a horizontal or vertical line or concentric rings. In all of these cases, the point is that, this is just to give you a flavor of the different conditions, but in all cases, we kind of have a continuous set of targets, and that's going to be important. In fact, that's what I'm going to emphasize on this slide right here. So imagine what's happening as the monkey is about to reach to position X. So the screen that the monkey's looking at is telling it where to execute the reach. You can imagine that what happens if the monkey is reaching to X plus or minus a small epsilon. As epsilon goes to 0, the activity on average in motor cortex should converge to the same thing, is the basic idea. So our assumption, or prior, if you will, is that the neural activity is going to smoothly change as the motor target changes just a little bit on the screen. So moving back to this firing rate space where each axis is the firing rate of a different neuron, we can think of the mean activity in motor cortex right before the animal is about to reach to one of these 3 targets. And we can extrapolate this out to reaches along the full ring that the monkey is making reaches to. And our assumption is that assumption or what we see empirically is that the neural activity is going to change as a smooth function as you smoothly vary the target. Okay? Now, taking this a step further, what I told you is we don't only care about what is the average activity in motor cortex, but we care about what is the level of noise in that neural population. There's actually substantial noise empirically in neural population codes from trial to trial. So what we are interested in doing is not just estimating this mean response, which kind of traces out a smooth ring in firing rate space, but at each point on this ring, we want to estimate a covariance. And just as the mean response, or the first moment of this distribution is changing smoothly, we want to have a model where the second moment, or covariance of this distribution, is also varying smoothly. This is a broadly applicable idea. I just walked you through it in a motor cortex example, but it would also apply to the example I was telling you about at the beginning with discriminating a visual stimulus. And there's many other applications in neuroscience, such as discriminating odors, and et cetera, et cetera, et cetera. OK, so very quickly on the method, I've kind of already told you that we're going to be using Gaussian processes to estimate the mean response. OK? And you've already heard this at this point from the previous 2 talks. But the basic idea of a Gaussian process is I'm going to put a prior over nonlinear functions. The prior is going to be specified by some kernel with some length scale and parametric form. I'm not going to go into those details. But then what I do is I condition on observations. Now here the observations are in a model where we're assuming very little noise. But the idea is essentially the same. So we're going to use this Bayesian inference machinery to basically do a fancy form of nonlinear curve fitting. And that's the first step of the model. The second step, which is the more methodologically novel contribution to the neuroscience literature, is to extend this to estimate not just the mean, but the covariance. Now, to do this, we use a generalization of a Gaussian process, or somewhat of it, you can think of it as a generalization of a Gaussian process, I guess, called a Wishart process model. Some of you may have heard of a Wishart distribution before. A Wishart distribution is a way of putting a prior or specifying some other distribution over a set of positive definite matrices, covariance matrices. A Wishart process generalizes a Wishart distribution so that the covariance matrices are actually a function of something, in this case, the stimulus x. So very quickly, the high-level idea of how you construct a Wishart distribution is you use Gaussian processes. So in this case, I have a set of conditions x. That's going to be one dimension of the three-dimensional array I'm going to build up. I have n neurons. This is, remember, a high-dimensional space. So this is number of dimensions. And then because we're in a very high-dimensional space, I want to specify a prior that favors simpler covariance matrices. Maybe I don't care about getting the full, like every dimension correct on this ellipsoid, but I really care about the top principal components or the top dimensions that explain the most variance. So I'm going to put a Gaussian process prior more or less on the top principal components of this covariance matrix as a function of the condition, as a function of where the monkey is reaching, or as a function of the visual stimulus I'm showing you. And then that's basically it. I just use this tensor u, which is a function of stimulus x, to compute a covariance by basically saying u transpose. And then I'm going to add a diagonal term so that the whole thing is invertible. You can extend this so that you can make this higher and higher dimensional by adding more and more principal components, and then eventually this diagonal thing becomes less and less necessary. But this would be a nice number of parameters that we can actually fit to data. And it's basically a low rank plus diagonal prior on the set of covariance matrices. Okay? Yeah.
So, Alex, I had a question. Is this covariance matrices or precision matrices? Because I'm
It's a
used
good
to
question,
Wishart's going along with precision. So.
It's true. Yeah. So I think it's you can do it either way, but there's a lot of numerical stability issues if you use a inverse Wishart prior. And also, conjugacy is out the window in all of these models anyway, so it's not clear to me that the inverse Wishart would give you the same computational advantages in the simpler cases. But to be honest, it's something that we played with a little bit, and it wasn't working. And then, but yeah, we are actually specifying the covariance, not the precision matrix, is the short answer. Other questions? I've been kind of just barging through all this. Yes, Jeff.
Just so I'm clear here, so you're assuming continuity as the target moves continuously around the circle.
Yeah.
Is there any influence of the muscular control involved? There might actually be discontinuities in the control signal, or is that just outside the scope of this
No, I don't
task?
think. Yeah, there shouldn't really be discontinuities. So remember, the idea is okay. The monkey is about to make a movement. We're just looking at that snapshot in time. There is a whole nother story here, which is thinking about how the dynamics are unfolding over time. But we're just looking at a snapshot of time. And the intuition for why things should be continuous is just like in the first slide I showed you. The monkey can't see perfectly where the target stimulus is. So if I moved, in other words, if I moved the target reach condition by a fraction of a millimeter, that's going to look the same to the monkey. So across trials, the neural activity should also look the same. So everything gets smooth.
About some targets require pronation or supination of the forearm or whatever. Don't worry about that.
Don't worry about that. That is an interesting point, though, that maybe we would assume that the length scale should be non-uniform across the ring, because at certain locations the complement which set of muscles are being activated is changing at a faster rate. That's a great question. We haven't looked at changing the length scale as a function of x, but that would be interesting. Okay, so I guess I'm kind of taking questions as I go. I'm somewhat running out of time, but okay. So very quickly, the punchline to this talk is that Gaussian processes work well, and Wishart processes also work super well. So this is what happens if I this is a simulation, but imagine that the monkey is making reaches along this ring, and I just independently try to fit the mean and covariance to each condition, and I don't do any pooling of statistical power across neighboring conditions. This is kind of intuitively very suboptimal, but it's what everybody in the field does. So we're trying to fix that by showing, you just get very noisy and bad estimates. If you use a Wishart process and a Gaussian process model, you can pool statistical power from the neighboring conditions, and then you match the ground truth, right? That's all this plot is showing. And you can push this very far and actually sample conditions along the ring and only have one trial per condition. But you can still, with this prior distribution, get a good estimate of the marginal noise in any individual condition, again, through this pooling operation. So, if I were to try to fit a ellipse to this training data, it would be impossible because I don't even have two trials from the same condition, so I have no estimate of any variance. So by building in the Wishart process prior, the idea is that you can leverage information from nearby conditions and still get a good response.
Excuse me?
Yeah.
Yeah. Doesn't the Gaussian process already give you a covariance information? I mean, why are you augmenting that with this?
Sorry. Yeah, the Gaussian process will give you a covariance on the posterior over the mean responses. I'm actually not even showing posterior estimates here. You can think of what I'm talking about as a heteroscedastic regression, just to put other names on it, where at each value of x, I want to predict what is the outcome going to be, but also a measure of the variance in my dependent variable. So that's fundamentally different than what the Gaussian process gives you. The Gaussian process covariance is how these different points covary with each other, but it doesn't tell you how much variance there would be at a single condition from trial to trial. Does that make sense? There's kind of two notions of noise here. I guess noise in my estimate of the ring, and then noise of if I tell you what point on the ring, what condition I'm in, what's the trial by trial variance. Okay, great. I have 5 minutes left. I mean, the punchline of this is that this works really well on real data. I want to spend just a couple minutes talking about a cool recent collaboration with David Brainerd at UPenn, who was visiting here, and his postdoc, Fangfang Hong, where we apply this to behavioral measurements, not to neural measurements. So David is an expert on color vision, and he puts humans in a kind of phone booth and makes them perform thousands of trials that kind of look like this. You show 3 color patches and then ask the person to report which of the 3 color patches is different than the other 2. And again, you can make this arbitrarily hard. So hopefully this is a case that you probably can't tell which of these shades of green is different. This one is the different one. What's really interesting about this is if you look at people's ability to discriminate colors, it really depends on the base color. So for white or gray, if I just change the color a little bit, you'll be very easy to detect, or very quick to detect any change. Whereas if I look at the edges of RGB space, the resolution gets, your ability to perceptually discriminate color changes is much worse. Okay? So this is another example where where we get these elliptical fits to local discrimination performance, and we believe that these ellipses are changing smoothly as you vary the stimulus. And you can fit these ellipses not to neural data, but to human behavioral judgments. So the classic way of doing this is you start with some reference color, and then you march along some dimension in color space, and then find the point where you start to be able to detect the color change with 75% accuracy or something like this. Okay, so I would fit that, and then I would look at a new direction in color space, and I would determine the threshold in that direction. And then I would do a third direction, and then et cetera, et cetera. I build up an estimate of what the local color discrimination is, and then I would fit an ellipse to these points. This is how McAdams and more or less how McAdams did this with others back in the 1930s. This is very, very labor intensive. It takes like thousands of trials just to fit one of these ellipses. And then you have to go do it again. So you start with another reference color, you go in one dimension, another, et cetera, and then you build up another ellipse, which is going to have a slightly different size and a slightly different orientation. Okay, and in fact, this is so hard that people have only done this through 2D slices of the color space. Nobody, to our knowledge, knows what this looks like in the full 3-dimensional 3D color space because people think it's too hard. In fact, there's this really fun quote by Erwin Schrödinger, who studied this problem a lot. He was very interested in color discrimination, where he basically says this is completely hopeless, it's hopelessly difficult. And I just couldn't, I had to show this slide because I have a little bit of an identity crisis, whether I'm a neuroscientist or a statistician. But these days I'm kind of thinking, maybe I'm a physicist, like Jeff Hinton is a physicist, that sort of thing. So who knows? Okay, so I'm running. Oh, it's RGB. So we have 3 different photoreceptors, so it's like a 3-dimensional signal in our retina. Yeah. Okay, so I'm 30 seconds left, so just going to tell you the punchline here is that you can build up a probabilistic model using Wishart processes with a Gaussian noise assumption under the hood, and fit these fit these performance discrimination threshold contours using a Wishart process. And the punchline is that similar to the neural data, this works really well. So this is, if you kind of match the number of trials you will give the model, this is the traditional way of fitting these so-called macadam ellipse contours. If you use our method, you can do much, much better even with fewer trials. So this is simulated data, but David is gearing up to apply this to real measurements that I think Fong and other people in the lab are going to sit in a closet for a week and try to map out the full 3D space. So very exciting. So what I try to tell you in this short talk is, one, that neuroscientists are really interested not just in like the mean input-output response of a neural circuit, but studying the scale and the correlations of noise across neurons. Estimating that noise covariance is extremely challenging, especially in high dimensions. And I introduced this idea of a Wishart process, which is, kind of close cousin to the Gaussian process, which has proved to be very useful in neural data. And also, we're applying this now to human data as well. And with that, I'll thank my lab and take questions.
All right, we had a lot of questions along the way, but time for one or two, maybe? Yes.
Is it one hypothesis when it comes to like motor control, that the noise covariance is one thing that can allow you to let's say you've learned a specific motor behavior. How do you then go from that learned behavior to something that's slightly different or very different?
Yeah, okay, so I think I know what you're referring to. There's a paper that says in a brain-computer interface setting, if I give a monkey a new task or a new mapping between the neural activity and the way the computer cursor moves, there are some configurations where the monkey can learn it really quickly, and there's others that can learn very slow. So yeah, we haven't applied this methodology to that data set. I mean, I think there's only maybe a couple data sets out there that would even speak to that question. But I think it's potentially related. Byron Yu, the person who published that, is going to be coming in June as part of like a conference that we're holding. So it'd be fun to ask him.
Yeah, I think they've done some similar stuff with mice as well, where they have
Okay.
they learn to reach to one direction, or they learn to reach a specific type of task.
Yeah.
And then they have to learn a different direction, but they know the overarching behavior, but then how do you go from that locked in stereotype behavior?
Yeah. Yeah, I mean, I think there's only a couple papers on this general idea, and a lot of, I guess I didn't say it too much, but my personal view is that it's really difficult to statistically estimate a lot of these things. So yeah, we're excited to look into that and hopefully use more refined tools to really test and verify these ideas.
All right, let's thank Alex once again.